So, good morning, students.
Good morning, sir.
So, it took some time to start this class because of some technical issue. So, I restarted my laptop and now it's on. Okay. So, we'll continue with uh, the topic which we were doing yesterday. So, yesterday we were talking about serum, right? We were talking about the technique of how to withdraw blood. So there was a lot of discussion, we had a lot of points. So students, if you have any question from that, you can ask and then we can move ahead. Any questions? Okay, so let me ask a question. What is anti-serum? So students, just raise your hand. What is anti-serum? So do you remember we talked about anti-serum? So I can see one hand now. Yes, Yukta, what is anti-serum?
Sir, anti-serum basically is uh, serum containing antibodies, which is uh, extracted from immunized organ.
Extracted from immunized organ?
Which is organism. I said organism, not
Okay. So yes, you're right. It's the serum which is containing antibodies. The only thing is here we are talking about a specific antibody against a specific antigen. Okay. Just for example, snake venom antitoxin. Okay. So it's basically the serum having antibodies, right, against the snake venom. Okay, so that is antitoxin. Okay, okay. Next is what is the difference between serum and plasma? Who will tell this? I think this is very easy. Yes, Devanshi.
Sir, serum is the liquid that remains after the blood has clotted, whereas plasma is the liquid that remains when clotting is done or prevented when we add anticoagulant.
Okay. So does plasma have the coagulation factors?
Yes, sir.
yes, sir.
Okay. So students, what we can do is we'll first start with the difference between serum and plasma and I'll show you a small video on it. Okay. And once we are clear with the differences, we'll move ahead to the steps of serum separation. Okay. So let's see this video first.
Blood can be divided into two main components. The formed elements, that is cellular portions, and the blood plasma, that is the fluid part of the blood. The 45% of the cellular portion consists of RBC, WBC, and platelets, whereas the 55% of the blood plasma consists of water, proteins, and other solutes. The proteins mainly consist of albumins that regulate oncotonic blood pressure, fibrinogen that is a clotting factor, and globulins that are antibodies. Plasma and serum both are derived from blood. Upon centrifugation of blood in the presence of anticoagulants, RBCs settle down as they are most dense, followed by a buffy coat of WBCs, followed by plasma, which is the least dense. Since anticoagulants are used, the supernatant contains clotting factors like fibrinogen. On the other hand, to obtain serum from blood, the blood is allowed and platelets, whereas the 55% of the blood plasma consists of water. 55% of the blood plasma consists of water, proteins, and other solutes. The proteins mainly consist of albumins that regulate oncotonic blood pressure, fibrinogen that is a clotting factor, and globulins that are antibodies. Plasma and serum both are derived from blood. Upon
Okay, just hold on. There is some issue in this.
plasma and serum, blood can be divided into two main components: the formed elements, that is cellular portions, and
Okay, so I'll just stop here to understand the difference between plasma and serum. Okay, so you can see when we just do the centrifugation directly with anticoagulants, right? So we are using anticoagulants here, okay? So the RBCs settle down, right? And whatever left above is your plasma, okay? So in this, clotting factors, proteins, for example, fibrinogen are present. Okay, whereas serum is plasma minus the clotting factors. So plasma have the clotting factors. Okay, whereas serum it does not have the clotting factors. Clear?
Yes, sir.
And the use of plasma or serum, so why you need serum and why you need plasma? So we need plasma, basically it is given to patients lacking blood cells, okay. So uh, plasma is basically given to the patients, right? Whereas serum, it is usually used for the diagnostic purpose, right? So we have already talked about the importance of serum. So uh, if there are some tests to be conducted, right? Just for example, some diagnostic test, uh, if you have to carry out ELISA, if you have to carry out any other specific diagnostic test, you will use serum. Okay, so we usually don't use plasma for such diagnostic tests. Okay, so I think now it's clear. So serum is plasma minus clotting factor, and plasma is the one which is containing the clotting factors. Okay. So next is how we can separate plasma or serum in the laboratory. Okay. So there are different steps. So again, I will show you a video. Okay, that video discuss the steps, so you can actually see what is being done. Okay, and how after uh, the process is done, how this serum components look like. Okay, so let me share this video.
Dr. Edward Matthews, I'm here at Montclair State University in the Exercise Science Lab to show you how to take a blood sample and either turn it into a serum or plasma sample that can be either used or stored. So anytime you're handling a blood sample or other similar biohazard, make sure that you're wearing the appropriate clothes. So when you are working with a biohazard like blood, you need to make sure that you're protecting yourself from that biohazard. So you need to be wearing shoes that are closed so that there's no exposure of your feet. You need to be wearing pants that prevent any exposure of your legs. You should be wearing some sort of lab coat that protects most of the rest of your body. Then you need to be wearing gloves that protect you from anything that's going to get spilled on your hands. And you need to be wearing some sort of protective eyewear that's going to have some side.
So students, is it clear? So whenever you are working in a diagnostic lab, right? So this is the kind of precaution that is required. Okay. So as you just heard, shoes are important because shoes will cover like whole of your feet, right? If you are wearing slippers or sandals, then there is a chance that the blood or a drop of blood may fall on your skin, okay, on your feet, okay. A lab coat, I think you all understand. It again, it's basically a covering, right? So that if there is some spillage, or if suppose there is spillage of blood, so it does not fall directly on your clothes, right? Uh, you need to have gloves, right? And to protect your eyes, there are kind of goggles which are available, so you can use them. Okay, because see what happens sometimes uh, the blood drop may splash, okay, and if there is some kind of splash, the drops can then get into your eyes, right? So this kind of protection is usually required.
Shields as well as a lot of coverage to your eyes to prevent any splashes from getting to your eyes. So the type of blood samples we're talking about today are going to be collected either through a typical venipuncture, just using a needle, or an IV catheter, which is also a type of venipuncture. Either way, we're going to be collecting tubes that are typically going to be either purple top or red top. The purple top are the EDTA tubes that are going to prevent the blood from coagulating. The red tops are going to be the silicone-coated tubes that are going to promote coagulation of that blood. So whether you prevent coagulation or promote coagulation is going to determine whether you get plasma or serum. All right, plasma is what you get from the purple top tubes where you prevented the coagulation, so it still has platelets in it. Serum is what you're going to get from the red top tubes where you promoted coagulation. So essentially that coagulation is the clotting of those platelets. And so when you spin this down, you're going to end up getting just liquid and no platelets inside that liquid. So either way, you're going to take the blood tube when you first get it, and you're going to invert it very gently and very.
Okay, so the first thing is, students, you should know what you are looking for. Do you want plasma? Do you want serum? Okay, and accordingly, you can put or place the blood in that specific tube. Okay, the collecting tube or the vacutainer. So you can collect the blood in that according to your requirements. Okay. Uh, okay.
Slowly, typically between 4 and 6 times. And that's going to make sure that the blood has been thoroughly mixed with whatever type of treatment is inside that tube. Again, either a coagulant or an anticoagulant. And
okay, so I don't know if you have seen or not. So whenever you go to a diagnostic lab and you give your blood sample, right? The technician, after collecting the blood, right, they will mix the vial, or they will just shake the vial, right? They may shake like this, but this is not the right way, right? So they should invert it, and it should not have jerks. Okay, it has to be done in this way, right? So it does not have jerks, but the purpose is that suppose you have added some anticoagulant. So many of the anticoagulants are in the form of powder. Okay, so when you mix it like this, right? So what happens? There is proper mixing of the anticoagulant with the blood. Okay. Otherwise, the coagulant is just anticoagulant is just at the bottom of the tube, right? So if you mix it, then that anticoagulant mixes well with the blood. Okay.
Then you're going to take the blood and you're going to set it upright in a tube holder and allow the blood to clot if it is a coagulating tube, or you can spin it right away if it's a non-coagulating tube. All right, so one mistake I made that you're going to see in this video is that I forgot to put them in a tube holder. It doesn't really make a big difference, but it makes it hard to tell whether or not the blood has been coagulated, because how you tell if blood has been coagulated is you take it and you very gently turn it upside down, and if the blood doesn't
Okay, so in this picture you can see that the blood has coagulated. Okay, it has become hard, right?
move, if it doesn't flow downward, then that means it's coagulated. And you can still see that with it being on its side, it's just not quite as clear. All right, so once you have confirmed that your coagulated blood, or the blood that you want to coagulate, has coagulated, then you can take the blood samples and you can put them in a centrifuge. All right, centrifuges can be set to several different parameters. You can set the temperature of the centrifuge. So you can make it room temperature, which is approximately what I'm going to be setting it at, or you can make it cold, which oftentimes needs to be done if you're running a sample for hormones or something that's going to degrade quickly at room temperature.
Okay, so again, it depends. So in diagnostic labs, usually you won't see such kind of centrifuge. They're high-end centrifuge which can maintain the temperature. Okay, so usually the centrifuge that you will see in most of the laboratories, it will just manage the speed. Okay, you can see this speed button, right? So this is 1000 rpm, 2000 rpm, 3000 rpm. So this 1 2 3 4 is the rpm, or the revolutions per minute. Okay, so how many revolutions will it take, the centrifuge tubes will take in a minute? Okay, so usually for serum separation it is around somewhere around 3000 rpm, right? And in research laboratories you will usually see these kind of centrifuges which can also maintain the temperature inside this chamber. Okay, so just for example, if you are doing a specific test, okay, if you are looking for a specific protein which degrades very soon at room temperature, right? So in that case we usually use these centrifuges which can also maintain the temperature. So we set the temperature, so it could be 4 degree, 8 degree, whatever temperature you are looking for. Okay, and in that case this chamber, it becomes cold. Okay, so we are basically talking about the temperature inside this chamber. Okay.
The other things you can set are how fast it's going to spin. I'm going to spin it just beyond 3000 RPMs. Or you can set also the time of the spin, which is something that typically is between 10 and 15 minutes. And you can decide whether or not to have a brake applied, or how heavily to apply that brake at the end of the centrifuge time period. Set the those parameters.
Okay, so he also talked about the brake. Okay, so usually we don't apply brake, right? Because see, uh, the blood is in motion, right? Now, if you suddenly put brakes, so this momentum gets disturbed, right? And whatever has separated after centrifugation, that may get disturbed, right? So usually we allow it to settle down by itself, right? So that whatever centrifugation has taken place, it does not get disturbed, right? So it's up to you. You can always standardize if brakes are required, because see, if you apply brake, you can open it early, right? If you are not applying brake, it will take some more time to slow down and stop. Okay.
Parameters to whatever type of analysis you're going to be doing, and then you're going to put the blood tubes inside the centrifuge. Now make sure that you're putting it inside the.
Okay, students. So here you can see there are different types of uh, centrifuge holders, tube holders, right? So you can see, first thing, it has to be balanced. So students, whenever you are working with a centrifuge, all the four ends have to be balanced. Okay, so you can see this light green and light green, right? They are in front of each other. Okay, so they are balancing each other, and similarly this and this, right? Dark green and dark green. So they are in front of each other. They are balancing each other, right? Another thing, anyone, he has put this tube here. Is it okay? Is it okay to put this tube here?
No, sir. I think he should put it in the smaller ones.
Yes. Why?
Maybe because it is not fitting it in properly.
Yes. So if you put it here, and now just imagine if you start the centrifugation, this will keep shaking, okay? And proper separation will never happen, okay? So what you need to do is take this tube, put it in this place, okay? And another thing, is it okay if I just put one tube here and centrifuge?
No, sir, you'd have to put another tube in the opposite one in order to balance it out.
Yes. But suppose you just have one sample, then what will you do?
So you can just put maybe like a tube of water or something. It doesn't necessarily have to be another sample. It just has to be the same weight.
Yes. So so if you get into such kind of situation, right? If suppose you have one sample or three sample or five samples, then you need extra tube to balance it. Okay. So you can just put water. Okay. Now there can be sometime issue if the solution you are using, for example, if blood has high density, right? So it may have a bit more weight than the water sample, right? So in that case, you can put few more drops of water to balance it out. Okay, so we usually don't go for the exact weight. Okay, the machines are capable of balancing a bit of up and down, right? But it should be almost around, the weight of both the sides should almost be equal. Okay, so
The proper tubes within the centrifuge. So if you put it in tube holders that are too large for the tubes, it's not going to hold it very well, and you don't want that. So put it in the right size tube holders for the tubes you're using. All right, so the other thing you need to do when you're putting the tubes inside the centrifuge is try to disperse them in such a way, or to place them in there in such a way that they are balancing each other in their weights. So if you have one over here, have one over there. All right, and so that when it's spinning, it's going to have the same amount of weight on both sides of the inside of that centrifuge, and it's going to promote an even spinning. All right, so once the tubes are in the centrifuge.
Okay, students, one more thing, you can see this centrifuge. So these are called swing buckets, right? Why swing bucket? Because so I don't know how to explain this. Okay, now suppose this is your centrifuge bucket, right? Now what happens when this centrifuge will start, right? This bucket, because this is swing bucket, okay, this will move like this. Okay, so if this is the center, right, now your bucket is like this. Okay, so when you turn on the centrifuge, this will fly like this and this will move in this way, right? So this is swing rotor bucket. Okay, some of them are fixed, right? So they are in this angle, okay, and they will only move like this, okay, so they cannot swing. But the one which you are seeing, they are swing rotor bucket. So usually they are like this, but once the centrifuge is on, it starts moving. They will just fly off like this and move, okay. And if you apply brake, they will suddenly come down like this, okay. But if you just allow them to slow by themselves, then they will gradually come down and stop like this, okay. Clear?
Fuse appropriately, press the lid down until it clicks, move over the locking mechanism on the lid, and then you're simply going to turn the centrifuge on if you haven't already, and hit the button if there is a button to start the spin. So this particular centrifuge does not have a button to spin. You just turn the timer to whatever you want it to be set at, and that's going to initiate the spinning process. So you're going to see a very slow increase in the RPMs of the device, because it's going to it doesn't want to start super quick.
So this will go up to whatever you have set here. So in this case, you have set it around 3000, right? So again, you can standardize. What you can do is 3000 at a particular time period. Okay, just for standardization, some people increase uh, the time, increase the speed and reduce the time, right? Or you can increase the time and reduce the speed, okay? It all depends at what time and speed you are getting the best outcome, okay?
Quickly, it's going to sort of ramp up,
So
and that's
this
going
will
to
now
prevent
reach up
too
to
much
3000
jostling of the
rpm.
blood inside the tubes, which is something you don't want. You don't want the tubes being sort of jostled around, because it's going to increase the likelihood of damaging the sample. All right, so once the centrifuge is done, it's going to start to break itself. If you have the brake applied, you can see that the RPMs are going to slow down fairly quickly. If you don't have the brake applied, it takes a very long time for the centrifuge to stop. All right, so once the centrifuge has stopped, you can open it. So you're simply going to flip the switch back on the locking mechanism on the lid, and then there's a button on the front of the centrifuge that you.
And the students, I'm sure you have seen those semi-automatic washing machines. There is a spinner in in that machine, right? So what we do is we sometimes just open it before it has fully stopped, right? So you can do that in case of clothes, but in case of samples, it is not advised to open before it has stopped, right? Because if you will open while it is still revolving inside, right, so it will stop with a hard break sometime, right? So in that case, uh, the whole purpose of separating may get defeated, okay? So you need to have that patience. You should allow it to stop, right? Give it that full time. And again, it is also important to prevent any damage to the centrifuge itself, okay, because these are expensive centrifuges. You get very small centrifuges in the lab. So whenever you will visit lab, I can show you those very small centrifuges. Uh, you can see a centrifuge which only have speed, no other thing, no temperature, no time, okay. But there are high-end centrifuges which, so like this one, it can maintain temperature for you. It will automatically, it can do the time, right? So if you just set it at 10 minutes, so it will end after 10 minutes. So you can set the time in this and you can set the RPM, right? So these are the normal centrifuge. Beyond this, there are ultra centrifuge, ultra high speed centrifuge, right? So in those centrifuge, the RPM can go very high, okay, and if you are looking for very small components, right? So if you want to settle down some viruses or something, then you may go for ultra high speed centrifuges, okay? So again, they are very expensive. You won't see them in the normal laboratories, okay? But you will just see these kind of centrifuges in the labs.
You have to push to actually open the lid. So there's two different locks here. So once both those locks are undone, you can lift the lid, take the samples out, and you can use them however you plan on using them. So when you're taking the tubes out of the centrifuge, and so when you're taking the and you can use them however you plan on
So
using them.
students, now you can see the blood after centrifugation, right? So all the RBCs, they have settled down, okay? And because you have added, if you have added uh, coagulants, okay? So you will have this plasma, the serum, okay?
So when you're taking the tubes out of the centrifuge and to wherever your workstation is, or when you're working with them at the workstation, make sure that you never invert those tubes again. So if you invert the tubes, you're going to see that the blood remixes partially, and essentially you're ruining your.
Okay, so you can see once centrifuge is done, you have to handle those tubes very carefully. ठीक है, ऐसा नहीं कि ट्यूब्स लेके तुम डांस करते हुए अपनी लैब तक जा रहे हो या वर्क स्टेशन तक जा रहे हो. Okay, you have to hold them with very stable hands. Okay, um, because he has already shown you if it gets mixed, then you can see the serum here, it is not yellow now, okay, and all the blood or the blood components, it has mixed with the serum, right? So for diagnostic purpose you cannot use this kind of sample, right? Sometime what happens, you when you are working in a lab and you get these kind of samples, so those samples are rejected, okay. People will not put these kind of samples in their machines, right? uh, so again, uh, after this step, centrifugation, you have to carefully work with the sample, right? If you have to remove this serum with the autopipette, you have to do it very carefully, okay? Otherwise, uh, specifically at this point, otherwise what happen, you will also pick some of the blood cells, okay. Okay, so question till this point.
Sir, am I audible?
Yes.
So I had a question. It's actually when you were explaining the differences between serum and plasma. How is plasma used for patients who have low red blood cells? Since I mean, if you've separated it out, then how is it beneficial to them?
No, it depends which cells you are talking about. You are not talking about RBCs, okay? In this case, we are talking about the WBCs, okay? Because if you have to give RBC, then you need to give the whole blood, okay? The blood transfusion,
Okay, okay. Thank you, sir.
okay? Okay, so let's see further.
So if you look at your tubes after they've been centrifuged, there's going to be a clear separation between the blood cells at the bottom and the clearer liquid on top. So what that clear liquid is, it depends on the type of tube, whether it's a coagulant or an anticoagulant tube. So again, the anticoagulant tubes are going to have plasma in them, so that liquid's going to still contain platelets. The coagulating tubes are going to have serum in them because all the platelets have coagulated and been spun to the closer to the cells in the bottom, the red blood cells at the bottom. All right, so either way, you're going to take those tubes, you're going to very gently remove the top, so just kind of wiggle it back and forth until it's able to pop off. And then you're going to take some sort of pipette, so we're just going to be using a simple bulb transfer pipette today, and you're going to suck up that serum or plasma and transfer it into some sort of container to be used in a machine or to be stored in a freezer. All right, so when you're sucking up that serum or plasma, though, start at the very top. So start at the top of the plasma and slowly work your way down as the plasma is sucked up into the bulb pipette. So the reason you're doing that is if you get too close to the cells about midway down the tube, you're going to disturb those cells. And doing so is going to remix the tube of blood, and it's going to essentially destroy your sample. So start at the top very conservatively, suck a little bit of the serum or plasma, and continue to lower the pipette as you're sucking up serum or plasma, and stop when either you fill the pipette or if you get too close to the cells where you start thinking that maybe you might suck a little bit of the cells up and mix your sample. So stop before that happens. Then take that sample, put it wherever you're going to use, store or use it, and then if you need more, you can go back and try to get a little bit more serum or plasma, but you want to make sure that you get the easy serum and plasma first before you start getting a little more gutsy and getting the serum or plasma that's closer to the cells. So once you have transferred all of your serum or plasma to another container that you're going to use for analysis or for storage, you have officially aliquoted the serum and plasma. So if you're going to store it, make sure that it goes into a refrigerator or freezer more likely, and that happens quickly, because any time that the sample is at room temperature, it is degrading the sample. So make sure that you get it into its storage location as quickly as you can. All right, so that was a really quick overview on how to spin blood and pull off either the serum or plasma for storage or use. If you have any questions, please put those in the comments below and I'll try to get back to you. Otherwise, please.
Okay, so students, uh, I hope the steps are clear to you now. Okay, and one of the precaution is that once you have isolated the serum, okay, once you have used it, it should be stored at lower temperatures. Okay, so it could be at your like refrigerator, right, or -20. Okay, so if you are thinking for long-term storage, suppose you are a PhD student and you have to use that serum sample many times. So, or if you want to store it for a long time, just take that sample and put it in your -20. Okay, and so I'm not discussing those things, but yes, there are a lot of things that you will learn when you are actually doing it. Like, just for example, if you are writing on those tubes, okay, and putting it in refrigerator or -20, right? And maybe after a year you take it out and you may find that whatever you have written, it has disappeared. So it will be a problem for you, right? So there are a lot of ways by which you can permanently have that indication or that mark on the tube so that the water does not take out whatever you have written on those tubes. Okay, so those things are like when you will actually do, you will understand all those things. Okay, so any questions? Okay, so I think you keep thinking. If you have any question, you can ask, and in the meantime, I'll have your attendance. So students, be ready with your cameras. So we'll start with Adish. Adish, okay, today I'm going in the different order. Okay, so we'll start with Yuvraj. Yuvraj,
Present, sir.
Yuvraj, you are not visible. Okay, Yukta,
Present, sir.
Yukta, you are not visible. Vivek,
Sorry, sir, network issues. I'm present.
okay, Tridev,
Present, sir.
Tridev not visible.
Sir, I just turned on my camera. Am I visible?
Okay, Tanvi,
Present, sir.
Shivani,
Present, sir.
Shivani, you are not visible.
Present, sir.
Okay, Shakti,
Present, sir.
Sanjana,
Present, sir.
Sanjana, you are not visible.
So my camera is on.
Okay, Sahiba, okay, Rupal,
Sir.
Nihal,
Present, sir.
Namrita,
Present, sir.
Manaswini,
Present, sir.
Kshitij,
Present, sir.
Janvi,
Present, sir.
Ishita, Devanshi,
Present, sir.
Sam. Okay. Ayushi.
Present, sir.
Adish. Okay, students. So this was one of your practicals. So you can make a practical file of it. Okay. And maybe in a week's time you can send it to me. Okay. So I'm ending the class now. And if you have any questions, we can have those questions in the next class on Tuesday. Okay.
Thank you, sir.
Okay, everyone.